All right, so we are moving on with inner plant structures, and we're talking about roots, but we're going to talk about a little bit about roots in general. To begin with, kind of the structure roots, and we've talked about tap roots and fibrous root structures, et cetera, a little bit. But we're going to review that, look at some different types of roots. We're going to dive into the root, so inside of the root, and take a look at what the cell structure looks like, what those structures do, what the point of them is, and then how that relates to one of the major functions of roots, which is uptake of water and nutrients. Okay, so that's kind of where we'll finish off. So let's dive into plant roots. So of course, three different types of roots: tap roots, fibrous roots, and adventitious roots. So our tap root systems are typically, again, associated with dicot plants, and then our fibrous roots are typically associated with monocot plants. And then adventitious roots are some little different ones. There's always some little funky ones out there, and there's a lot of plants that have the ability to create and utilize adventitious roots for a lot of different things. So again, taproot, primary root created from the enlarged radicle of the seed, typically thick and grows downwards, with lateral roots branching off. And we've looked at this one before, maybe. This is an example of a taproot. Fibrous roots, on the other hand, usually formed by thin, moderately branching roots growing from the stem. Okay, again, this is primarily found in monocotyledonous plants and ferns. And one of the typical characteristics of fibrous root structures is that they grow very shallow, okay, in relation to taproot structures. Now, adventitious roots are a little bit different. So some roots called adventitious roots arise from an organ other than the root itself. Okay. Usually it comes off of a stem. Sometimes it can come off of a leaf, depends on the plant. They're especially numerous on underground stems. The formation of adventitious roots makes it possible to vegetatively propagate many plants from stem or leaf cuttings. Okay, so we utilize plants' ability to create adventitious roots to our advantage, and we can utilize these to propagate these plants. So let's look at some examples of adventitious roots. This is a ficus tree, and these are called buttress roots. So the main tree probably started somewhere in here. As it grew up, it actually starts dropping down some roots or some modified stems towards the ground until it hits the ground, and at that point it roots in, and that's considered an adventitious root. Now, the reason that this plant does this, probably better chance of genetic survival. So if something happens, it's rooted in a lot of different areas, as well as, given the overall structure of this plant, it could be for support as well. Now, another plant that has adventitious roots, which many of you guys are probably pretty familiar with. This is a very popular indoor plant, utilized a lot, it is a great plant, great house plant. Find the right spot for it, and you can have quite the specimen in a relatively short period of time. Now you can see this in a house, and you can see these adventitious roots popping down. Okay, it's looking for something to root into, and you can see it here, planted in landscape, couple ferns around it, and you see these adventitious roots popping down and finding their way to the soil, and ultimately they'll root in. Monsteras, this is Monstera deliciosa. these actually grow in trees. Okay, and so typically they're found in trees in tropical areas, and as they grow up a tree, they will start sending down roots, adventitious roots, to be able to gain some nutrition out of the soil itself. Okay, so from a propagation standpoint, we can certainly utilize this by essentially putting these plants, these roots into containers. We can get them to root in, and then once they've rooted in, we can uh, remove them from the mother plant and then grow them out. So what are these adventitious roots? The ones that we typically see, and this is a picture of a plant that in our region, in the Sacramento region, this is not a desirable plant. This is Bermuda grass. Okay, and adventitious roots, Bermuda grass uses both of them. So rhizomes and stolons. Okay, so on the top picture here, we see a picture of rhizomes. A rhizome is a continuously growing horizontal underground stem that puts out lateral shoots and adventitious roots at intervals. Okay, so the rhizome is technically a stem. It's a modified stem that grows underground. This right here is an adventitious root that's coming off of the stem. Remember, if we pop back here, adventitious roots arise from an organ other than the root, usually a stem. Okay, and that's what this is. Okay, so we have an adventitious root here. We have a rhizome coming through here. The rhizome is a modified stem. A stolon is a creeping horizontal plant stem that is above ground. Generally, we find them right at the surface, but above the ground. And it can take root at points along its length to form new plants. So these stolons will do the same thing. As it continues across the ground, at the stem nodes, it will create some adventitious roots, and it will root in. Okay, I'm gonna like jump off really quick here and look for forsythia. So rhizomes, stolons, and we're talking specifically about adventitious roots. And I should have put this in, but might as well throw it in now, right? So here's forsythia, okay, border forsythia, beautiful plant. The ones that are generally used, they are hybrids, gorgeous yellow flowers in the springtime, like just profuse flowers, as long as they're maintained correctly. And then a very beautiful shrub other than that. Now, you can see these stems popping up and out. The newer stems are the ones that actually bloom the heaviest. Now, forsythia is really good about creating adventitious roots. And so a lot of times when we're using plants like forsythia or propagating plants like forsythia, certainly we can take vegetative cuttings. They're very good about developing roots off of vegetative cuttings, but you can also do something like this, where essentially you just simply take the one of the stems, and you'd probably want to use a little bit of an older one because essentially first and second year growth on forsythias have the most profuse blooms. Anything older than that, they tend to sort of bloom out. Okay, they won't get as good of blooms going through because it blooms best on new wood. So if you have a forsythia, you can take one of the older branches. You can do it with the newer branches as well. But you can take one of those branches and literally just bend it down, set it on bare soil, put some compost, a little mulch on it with a brick on top of it. And literally a couple months later, as long as there's moisture, this is what you'll find. And then you can cut this portion off of the mother plant, and you have a brand new plant. Okay, so again, we use adventitious roots to our advantage in horticulture to be able to make more plants. Now, adventitious roots can be a real bummer in the landscape if you're trying to maintain a landscape in really good condition and you're somewhere like Sacramento and you've got Bermuda grass. Now, just like with the forsythia, as soon as these things root in, right, they will create new plants. Eradicating Bermuda grass can be a real challenge because if you chop it up, mow it up, try to dig it out, every single little piece of rhizome that has a node on it, will essentially become a brand new plant when it comes out of dormancy. Okay. Now, Bermuda grass has a bad name, especially in, and I guess I should warn you, I'm going off on a bit of a tangent right now, but it's always fun. So, Bermuda grass in the Sacramento region, really Northern California in general, has a pretty bad name because it tends to invade and take over other grasses, lawns specifically, other parts of the landscape as well. But so the reason that we don't like it is because it goes dormant in the wintertime. It is a warm season grass. And there's a big difference between warm season grasses and cool season grasses. Warm season grasses go dormant in the wintertime and they are green in the summertime. Cool season grasses are green in the wintertime and will go dormant in the summertime unless they have water. And so in California, because we have a moderate climate, we tend to think that we deserve to have green grass year round. And that's sort of gotten us into a pretty bit of a pickle with how much water we have to put on our lawns. We'll get into that more later, but to be honest, if you're familiar with Bermuda grass and you've fought it in landscapes or your lawn or projects or whatever, understand that actually one of the best sods that you can get and is used widely are hybrid Bermuda grasses. And what they do is they try to breed into the grass shorter dormancy periods so that it's less, or there's less time during the winter that the grass is actually dormant and dead looking. Okay, so anyways, I understand Bermuda grass can be a challenge. Don't hate Bermuda grass. Chances are over the next 2 or 3 decades, it's going to end up, our hybrid Bermudas are probably going to end up taking over the lawn market in California. So back to and out of my tangent, rhizomes and stolons are both modified stems. Adventitious roots are grown off of these modified stems. Okay, now let's look at some other rhizomes. So other ones that we're familiar with, bamboo, right? Bamboo is a really cool plant and very useful for a number of different reasons. But it can be a real challenge especially the really prized ones. This is black bamboo, and black bamboo is gorgeous, a great screen, really helpful and useful in the landscape, but incredibly aggressive when it comes to its adventitious roots, which again, with these, they're arising from rhizomes, so the underground modified stems. And you can see how that happens. Okay. Now, right here, this is the solution to containing bamboo. Okay. This is the solution for containing the bamboo from traveling where or going into areas of the garden where you don't want it, or it's unintended. This is a polyethylene root barrier. You can see that it's overlapped here, and actually what they used is a steel bar on both sides. To be honest, I've done this before, and the bamboo got out. Okay, essentially, those rhizomes will find their way to the seam here, and then it will eventually get to that point between kind of where the steel plates are sandwiched, and it'll work its way through it, and it will bend it. So doubling that up is important, and then sealing the sides. Generally, that'll do it. Anyways, anyways, back to rhizomes and adventitious roots. So you notice that the trench here really only drops down 18, 24 inches. Okay. Now, do bamboo roots go deeper than that 18 to 24 inches? Well, remember, bamboo is a monocot. It's got a fibrous root structure, and so it's typically shallow root structure, but bamboo is a little bit different. Bamboo's roots will go to 18, 24 inches deep or deeper, but it's only the roots that are down seeking water. Okay. The roots that come off of rhizomes only hover within about the top 12 inches. That's why you don't have to go deeper with this root barrier. So it's those rhizomes that'll circle, and then those adventitious roots pop off of them. So if the rhizomes come out, as that rhizome travels along, it's going to have nodes. Remember, it's a stem, so it's going to have nodes, and at those nodes, we're going to find those adventitious roots. Some other rhizome cool things here. Hops. Okay, hops that's used in the floral industry and to typically flavor beer and other beverages. These are grown from rhizomes. So if you buy some hops, if you order some really specific types of hops, in the mail, you will receive rhizomes. Okay, so you're going to get a stem cutting, and that's what you plant. Okay, that's how you plant this. Now, very different than planting things from seed, right? Because you get that genetic variation when you're planting from seed. When you're planting using a plant part, you're cloning, and so you're getting a genetic replica of that plant. Strawberries, right? We've probably all seen this and taken advantage of it in that strawberries have stolons. Okay, so above ground, traveling above ground. And as those modified stems travel at those nodes, if it finds its way to some sort of rootable material, then it will grow. Okay, essentially, these are runners, stolen, and then these are daughter plants, but they are clonal replicas of the mother plant. A little bit more about that. So if we're talking about things like this, right? Well, here is technically, I guess technically, not really though, the oldest known living tree. 9,550-year-old Norway spruce tree called Old Chico. I think that's how you say it, but I'm not sure. Located in Sweden. It is the oldest known living tree in the world and has become a symbol of durability, sustainability, and regeneration. Regeneration. Okay, so if this is 9,550 years old, you should be able to take a cross section or a core sample through this trunk and be able to count 9,550 growth rings. Not going to happen though, because it's a clonal tree. So essentially, this tree has been here for a really long time, I guess specifically 9,550 years old, but it has fallen over. It has dropped branches to the ground, and adventitious roots have rooted in. So they have dated it to 9,550 years old, but it's not that this portion of the above ground tree that you see is actually 9,550 years old. It's just a clonal tree. Now, if we want to talk about the oldest trees in the world, which I like to, Pinus longaiva. This is the bristlecone pine. They're located in the White Mountains. The oldest one is over 5,000 years old. And yes, for actual tree that's not a clonal tree, these are the oldest living trees in the world, located in California. Very cool spot, beautiful trees. And of course, I'm on a tangent, has nothing to do with roots or anything. I just wanted to make it clear that there are clonal trees. We do get adventitious roots coming off of stolons and rhizomes and other plant parts, just like the forsythia. And so then I always like to squash the fact that, yep, it's 9,550 years old, but it's just parts of this plant that have aged that long, or the original plant did. It's managed to propagate itself through cloning. Whereas in California, we've got literally the oldest continuous living tree in the world. So go check these out if you ever get a chance. Now, let's go ahead and get into the roots themselves, okay? So if we look at a vertical cross-section of a root, we see 4 different zones. There's a zone of maturation, a zone of elongation, a meristematic region, and then a root cap, okay? So if we start sort of up in the stems, last lecture we finished at stems, right? And now we're going to start going down into the soil. If we go into the zone of maturation, okay, we get into essentially this portion right here. Okay, so it's this portion right here. Now, as this root goes deeper and deeper into the ground, okay, the root portion that was higher up in the soil profile becomes more and more mature, okay? As it becomes more and more mature, it starts to differentiate, okay? So you get some differentiation in here. So this zone of maturation, cells differentiate into different types of cells, okay? Here in the zone of elongation, we have the main goal of that root is to get deeper within the soil, is to find water essentially, and anchor that plant to the ground. Within the meristematic region, remember those meristem regions, it's stem tissue, meristematic tissue. This is undifferentiated cells, okay? So things are being created here. Through here, these in the zone of elongation, it's essentially adding to, up above it, it's adding to cortex, essentially like it's primarily pushing cortex there, and we'll get into what that is. And then below it, it's pushing epidermis. Okay, it's creating epidermis, which ends up creating at the tip a root cap, okay, which protects the meristematic region. So there's a very thick epidermal layer that protects this really important piece here, which is driving the root deeper and deeper into the ground. Okay, so if we start looking at the cross section here, we've got xylem and phloem, and essentially the xylem and phloem only exist within the zone of maturation and just a little bit into the zone of elongation. At that differentiation point, okay, within the maturation zone, we see root hairs coming out. Okay, now the root hairs, we'll get into root hairs. They are modified epidermal cells. We'll get into the why on that. Up above this, you will start to see some lateral roots coming off of the main root. This can be lateral roots coming off of the taproot, and sometimes it could be lateral roots coming off of fibrous root structures. Okay. Of course, you have the epidermis, works just like the epidermal layers above ground, but it's modified slightly so that it can actually take in water. We'll get into how that works. And then we have the meristem and then the root cap itself. So if we take a cross section, not a vertical cross section, and we look at this, here is our epidermal layer, okay, in red. And you see that this red goes around in this circle, and it also comes out here. This is a root hair. Okay, this is a modified epidermal cell called a root hair. Just inside of the epidermis, we have cortex. These are cells that are there for storage and the passage of water. Okay, so it's just sort of a buffer zone between the protective layer of the root, the epidermis, and then the endodermis. Okay, and this cortex is what allows water to flow through to get to the xylem. The endodermis is the innermost layer of the cortex itself, and there's some different types of cells within there that are called the endodermis. And within that, it does contain the Casparian strip, which is an interesting little part. Okay, it essentially, and we'll get to this, the Casparian strip essentially organizes all of the water that's coming through in a couple different ways through the cortex. And at this point right here, it says, I don't care how you got here, I'm going to make sure you get into the xylem. Okay, we'll get into what that is, but Casparian strip is a pretty interesting little part of the root cross section. It also controls what can go into the xylem. Okay, it controls what can go into the xylem. That being said, it's going to control nutrients that go into the xylem and anything else that's water soluble. Okay, so if anything makes it through there within the water, generally that Casparian strip within the endodermis is going to prevent it. If the plant doesn't need it, it's going to prevent it from coming out. Finally, we have the pericycle. Okay, so this pericycle here, the next layer in the purple layer, excuse me, that is the pericycle, and essentially it is there, and as this zone of maturation gets deeper and deeper into the ground, you can't see the pericycle here. The pericycle would be on the outside of the phloem. And as this root gets deeper and deeper and that zone of maturation gets longer and longer, it may decide to create a lateral root. And essentially the pericycle would modify to some degree and allow the zone of elongation to start heading out, and then the meristematic and the root cap to start heading out, and essentially create a new meristematic area. It would start pushing out, and then it would end up maturing to have all of this. Cross section here, just without labels, but this is what it actually looks like, not really the cartoon version. Okay. Now, mentioned root hairs. Okay, I mentioned root hairs. Root hair cells, the function is to absorb water and mineral salts from the surrounding area or surrounding soil. It's a modified cell. Okay, it's a single cell, and the root hair itself is just a modified portion of the cell. The reason that it elongates is to increase the surface area to volume ratio for faster absorption of water and mineral salts. Okay, so essentially there's these cells that the plant modifies. Okay, so those cells modify, and this single cell, by creating a hair that heads out into the soil, it drastically increases the surface area of the cell and therefore can take in water at a faster rate. This dude does a pretty good explanation on it, so let's check this out.
Hi and welcome back to Free Science Lessons. By the end of this video, you should be able to describe how root hair cells are adapted for the absorption of water and minerals. You should then be able to describe how water passes through the roots to the xylem. By the end of this video, you should be able to describe how root hair cells are adapted for the absorption of water and minerals. You should then be able to describe how water passes through the roots to the xylem, including the symplast and apoplast pathways. Okay, I'm showing you here the root growing from a seedling. As you can see, these roots are covered with very fine root hairs. Root hairs grow from cells in the epidermis of the root, in other words, the outer layer, and we can see that under the microscope. Now, water moves into the root hair by osmosis, and root hair cells are adapted so that osmosis takes place rapidly. Firstly, the densely packed root hairs massively increase the surface area to volume ratio of the root. Secondly, the surface of the root hair consists only of the cell wall and the cell membrane. This makes the surface extremely thin, increasing the rate of osmosis. Finally, the root hair cells use another mechanism to increase the rate of osmosis. The water in the soil contains dissolved mineral ions, for example magnesium, which plants use to make chlorophyll. However, the concentration of these mineral ions is lower in the soil than in the root hair. So root hair cells use active transport to move these mineral ions into the cell. The root hair cell also contains other dissolved compounds such as sugars. So because of this, the water potential inside the root hair cell is lower than in the soil. So water now moves into the root hair cell by osmosis down the water potential gradient. Now the water must move from the root hair cells through the root cortex to the xylem. Water can move through the cortex by two pathways. In the symplast pathway, water moves from the cytoplasm of one cell to the cytoplasm of an adjacent cell. To do this, the water moves through the plasmodesmata linking the cells. Remember that plasmodesmata are microscopic channels through the cell wall, connecting the cytoplasm of cells. The symplast pathway is driven by the water potential gradient between the root hair cells and the xylem. water can move through the cortex by two pathways. In the simplest pathway, water moves from the cytoplasm of one cell to the cytoplasm of an adjacent cell. To do this, the water moves through the plasmodesmata linking the cells. Remember that plasmodesmata are microscopic channels through the cell wall, connecting the cytoplasm of cells. The simplest pathway is driven by the water potential gradient between the root hair cells and the xylem. Water continually moves into the root hair cells by osmosis from the soil. This makes the water potential of the root hair cells greater than the cortex cells. In the xylem, the water potential is relatively low. So water moves by osmosis across the cortex, down the water potential gradient. Now I should point out that the simplest pathway is relatively slow. That's because the pathway for water in the cytoplasm is obstructed by the organelles. Water can also move by the apoplast pathway. In this case, water moves within the cell walls and the spaces between the cells. The cellulose cell walls have a relatively open structure, allowing water to move easily between the cellulose fibers. Remember that water molecules are attracted to each other, and scientists call this attraction cohesion. This is due to the fact that water molecules can form hydrogen bonds to each other. As water moves into the xylem and is carried away, more water moves along the apoplast pathway due to cohesion. The apoplast pathway offers much less resistance to water flow than the simplest pathway. Now, before the water can pass into the xylem, it must pass through a layer of cells called the endodermis. The cells in the endodermis have an unusual feature. A band of waterproof material called suberin runs around the cell wall, and scientists call this band the Casparian strip. Because of the Casparian strip, water can no longer move through the apoplast pathway. Instead, the water now passes through the cell membrane and into the cytoplasm, becoming part of the simplest pathway. By forcing all water through the cytoplasm, this allows the cell membrane to control which substances can enter the xylem. Now, cells in the endodermis use active transport to pump mineral ions into the xylem. This lowers the water potential of the xylem, triggering water to move into the xylem vessels by osmosis, and scientists call this effect root pressure. Now, you need to remember that root pressure is an active process requiring respiration. If we inhibit respiration by using a metabolic poison such as cyanide, then root pressure stops. Root pressure also stops if we prevent aerobic respiration by excluding oxygen. In the next video, we're going to look at transpiration.
Okay, so a couple things to point out there. Remember, going through this portion here, that water can come in through here. Of course, again, that root here is modified so that it can take more in. And he was talking about a couple specific things that we haven't necessarily covered. Okay. Now, the water potential gradient is one thing. Okay. Water will move from areas that have high water potential to low water potential. Now, that osmosis portion and that magnesium piece that this guy was talking about, essentially, water with salts in it, it wants to evenly, so the salts within water want to evenly distribute throughout that water, and the water wants it to evenly distribute it out as well. If you consider a large fish tank, if you dump a whole thing of salt into one side of that fish tank and agitate it, eventually all of the salt will uniformly disperse or distribute within that body of water. Eventually, it will be the same concentration of salt throughout the entire area. Okay, now the osmosis portion that he was talking about is osmosis happens through cell walls. Okay, so the root hair has magnesium ions in it, and it has a higher concentration of those ions within the root hair, since it's a higher concentration of magnesium within it, and there's a lower concentration of magnesium outside of it. What water does is water travels through the cell membrane to try to hit equilibrium, right? To try to uniformly have a uniform concentration of magnesium. Okay, so that's how the water gets in, and that's why that water is essentially sucked into that root hair through the cell membrane. Okay, now what he was talking about with the water traveling through, right? We've got the path of water through a root hair. The root hairs are there. It is not the only area of the epidermis that water can get through. Okay, it can also get through other epidermal cells. Once it's into the root itself, it finds its way to the cortex, and the cortex are sort of like different cells. Those cortexes are essentially they are there for storage and passage of water. And here we see the apoplastic movement of water between the cortex cells. And here we see an example of the symplastic passage of water through cells, okay, through cells to the endodermis, through the Casparian strip. And he mentioned suberin. Okay, the Casparian strip is a layer of suberin, a waxy material. Okay, and if we remember back to last lecture, Quercus suber, the cork oak, same thing is in that cork epidermis. Okay, suberin, it's a waxy material. So that is the path of water. Eventually, it makes it to the xylem, and as it gets to that xylem, at that point, it is going up the plant. Okay, and it's being encouraged through transpiration. Okay. Another little look. Oh, we already looked at this movement of water. There we go. Here's another like sort of vertical cross section of the path of water, water coming in through root hair. Of course, it can come between cell walls on the epidermis. And we got water movement coming through. Okay. And in this case, these arrows indicate the apoplastic movement of water. Okay, and this is primarily through cohesion. Okay, water wants to stick to itself, cohesion. And as water is in here, water keeps getting attracted to itself and keeps pushing in through the cortex. The symplastic movement, remember, comes through the cell itself and then goes from cells to cell through the plasmodesmata. Okay, the plasmodesmata. This is a way that water moves through the cortex, but again, as he mentioned, it moves very slowly because there's a bunch of stuff within the cells, whereas between cells in the cortex, you actually get more and faster water movement. Once both paths of water, right, reach the endodermis, it essentially hits that Casparian strip where it's brought into the endodermis cells and then through plasmodesmata and forced into the xylem. Again, apoplastic water molecule stays between cells in the cell wall region, never crossing membranes or entering cells until it gets to the endodermis. The symplastic movement of water is water molecules moving through intercellular connections called plasmodesmata, which are membrane connections between adjacent cells. Water from all paths must cross the endodermis. The endodermis is a layer of cells with a waxy inlay of mortar called the Casparian strip. The waxy inlay is the suberin layer, and that stops water movement between cells and forces all water through the same channel that leads to the xylem. Okay. And essentially the transpiration of water out of the plant, okay, that is the evaporative loss of water vapor from plant leaves through the stomata. Remember, in inner plant structures, when we're talking about leaves, you know, I'm pretty excited about stomata, what they do, how they function, etc. Well, as those things open and close and open and close, they are releasing water vapor. The intent is to release oxygen, right? But in the process, it's also releasing water vapor, which is a loss of water, but in that process, it's pulling water up and through the xylem, okay? And that works through cohesive tension and adhesive tension. That cohesive tension is the tendency of water to stick to itself, and the adhesive tension is the tendency of water to stick to other things. So let's look at this dude again. I like this guy's explanations. He's got a nice accent too.
Hi and welcome back to Free Science Lessons. By the end of this video, you should be able to describe how transpiration takes place in plants. You should then be able to describe the cohesion tension theory of water movement and the evidence which supports this theory. In this topic, we've been looking at how water moves in a plant. Remember that water is a reactant in photosynthesis, which takes place in the leaves. I'm showing you the structure of a leaf here. The main site of photosynthesis is the palisade mesophyll, which is found in the top half of the leaf. Although I should point out that photosynthesis also takes place in other parts of the leaf. Water passes into the leaf through the xylem vessels in the vascular bundle. Now, the surface of the leaf is covered in a waterproof layer called the waxy cuticle, and the job of the waxy cuticle is to reduce water loss from the surface of the leaf by evaporation. So as we've seen, water is an essential reactant for photosynthesis. However, photosynthesis also requires the gas carbon dioxide, and this carbon dioxide diffuses into the leaf from the external air. Photosynthesis produces the gas oxygen, which diffuses out of the leaf. Now, on the lower surface of the leaf, we find thousands of tiny pores called stomata. And when the plant photosynthesizes, these stomata are open, allowing carbon dioxide to diffuse into the leaf and oxygen to diffuse out. I'm showing you here a drawing of the internal structure of a leaf. Now, a key idea you need to understand is that the surface of the cells in the leaf are covered with a thin layer of water. This water evaporates from the surface of the cells, and because of this, the internal leaf spaces contain a high concentration of water vapour. Now, generally, the level of water vapour in the external air is relatively low, so when the stomata are open, the water vapour diffuses out of the leaf to the external air, and this evaporation of water, followed by the diffusion of water vapour, is called transpiration. Now, because of the continuous evaporation from their surface, the water potential of the cells in the leaf decreases. This causes water to move by osmosis from adjacent cells like this. This now lowers the water potential of these cells, causing water to move into them. At some point, this reaches the xylem, with water passing out of the xylem to adjacent cells like this. So when transpiration is taking place, water is continuously being pulled out of the xylem vessels, and scientists call this pulling effect tension. The movement of water from the roots up the xylem and out of the leaf is called the transpiration stream, and you need to be able to describe how this takes place. Now, we've already seen that water molecules form hydrogen bonds to each other. This type of attraction is called cohesion. Water can also form hydrogen bonds to molecules in the xylem vessel walls, for example carbohydrates, and this attraction is called adhesion. Now, one effect of cohesion and adhesion is that water can move up very thin tubes against the force of gravity, and scientists refer to this as capillary action. So when water is removed from the top of the xylem vessels due to transpiration, more water moves up the xylem vessels by capillary action to take its place, and this whole process is referred to as transpiration pull. So the combined effect of transpiration pull coupled with cohesion and adhesion is that water is drawn into the roots, moves up the stem and passes out of the leaves, and scientists call this whole process the cohesion tension theory. Now, even though this theory is over 100 years old, it is still accepted by scientists. There are two pieces of evidence which support the cohesion tension theory. Firstly, if a plant stem is cut, then air is sucked into the xylem, suggesting that the xylem vessels are under tension. However, the air prevents cohesion between the water molecules, so water movement stops. Secondly, if we measure the diameter of a tree trunk, we can see that this reduces when transpiration is at its maximum. This supports the idea that transpiration pull is generating a negative pressure or tension in the xylem. In the next video, we look at how to measure the rate of water uptake.
Okay, so he went through a couple things there that I had previously described, that water is pulled up and through, right? So the water moves through the root. Right? Water moves through the root, generally by diffusion. It makes its way through that cortex either apoplastically between cell walls or symplastically through cell walls. It hits the endodermis in the Casparian strip. It's organized and allowed to go into the xylem. Once it's into the xylem, there is that pull from the transpiration happening out of the leaves. Okay, so it is pulling that water up and through, up and through, up and through. Now, in a previous lecture, we talked about guard cells that create the stoma, and when the guard cells are turgid, the stoma is open. When it's flaccid, it is closed. Okay, so the amount of water that's available to the plant and the water demand of that plant generally will control how much water is actually coming through. Now, when there's not enough water in a plant or in the soil for the plant, we get wilting. Okay. Now, if we look at a rain event or an irrigation event, you after a rain event or an irrigation event, you get saturation. Okay, that is when all of the pore space within the soil profile that is adjacent to the roots or near enough to the roots that it affects the roots, essentially water fills all spaces in there, including all of the airspace. Of course, there's still the mineral and organic matter in there. However, all of the pore space is taken up by water. Generally after 24 hours after saturation, and this is soil dependent, okay? Some soils do this very quickly. Some soils take a very long time to do this. You hit field capacity, and that's generally once all of the saturated water that's filled up all the pores has moved through the soil profile through gravitational pull. Okay, gravitational pull. The water moves down and through, and you have adhesion of water to the mineral particles and the organic matter within the soil. So there's still water in there, but there's also air. Okay, this is a great sweet spot for plants to be at. There's air and water, minerals, mineral content, nutrients, etc. We'll get deeper into this later when we do a sort of shallow dive into soil. but this is field capacity, okay? And that is really when water is adhered to the particles, the solid particles within the soil. This again is sweet spot for the majority of plants. At some point through evaporation or transpiration, you get to a wilting point, okay? That is when typically wilting points happen when there's still water in the soil, but it is adhered so tightly to the mineral particles that the plant does not have access to it. Okay, it's essentially stuck so tightly to as a film to the outside of those mineral particles that it is no longer available to the plant. Now, that is when we hit a wilting point. Okay, now there's a temporary wilting point and a permanent wilting point. The temporary wilting point is the soil water content at which the plant wilts at daytime but recovers at night. The permanent wilting point is the soil water content at which the plant wilts and fails to regain life even after water conditions improve. Okay, there's been a turgor pressure loss. Okay, and that disrupts it. Okay, that disrupts it, and essentially at the permanent wilting point, the plant dies. Now, an example of a temporary wilting point, one that I like to point out, most people are familiar with hydrangeas. Okay, most people are familiar with hydrangeas. Big leaf hydrangeas are the most popular. They're beautiful plants. However, they are a high water use plant in the Sacramento region. So if we consider that it's a high water use plant and it has C3 respiration, C3 photosynthesis, where part of its transpiration process, it loses a lot of water through the water vapor through that stoma. So it's transpiring quickly, right? Now, what you see in the really hot part of the summer in the Sacramento region, next time it's really hot, just go out and look at some hydrangeas. Look at them during the day. If you know any of them that you can observe a couple times a day, take a look at them in the morning. Take a look at them in the afternoon. Generally, even if there's adequate moisture in the soil, it will be transpiring so rapidly that it can't keep up. And generally, you'll see when the water levels get a little bit lower, generally what you'll see is afternoon, those leaves will start to wilt. Once the sun goes down and it can recover, it will recover. And the next morning, those leaves will be perfectly fine. Okay, so that is an example of temporary wilting point. And then, of course, if there's no water there and it remains really hot, it'll get to a permanent wilting point. Okay, so that is it on roots. There's a lot to it, but again, in this class, we sort of hit high level, lightweight approach to botany, botanical structures, and the actual biology of it. But again, it is very important to have some general understanding of how plants work, what the parts are, what the functions are, how water moves through the plant. And then what we'll get into soon is nutrients, how nutrients move through a plant, and you know what problems can be associated with it. So that's it. Thank you guys. There's your lecture on roots.